glyceraldehyde 3-phosphate will enter the payoff stage of glycolysis. And the first step of the payoff stage is actually unique because this is the only oxidation reduction reaction of glycolysis. And since since here we have oxidation reduction reaction, NADH will be generated. So which carbon of a gap can be oxidized with some benefit for the cell? This will be aldehyde group of glyceraldehyde 3-phosphate. It will be converted into 1,3-bisphosphoglycerate. So you see here the oxidation state of this carbon is +1. It becomes +3. So glyceraldehyde 3-phosphate serves as a donor of 2 electrons, which will be transferred to NAD plus to form NADH. And here this phosphate group comes from inorganic phosphate. This mechanism, this step is catalyzed by glyceraldehyde 3-phosphate dehydrogenase. And please look at the details of the mechanism of this enzyme. So next, because this compound is high energy intermediate in glycolysis, and we remember from the list of rules that once a high energy intermediate in the course of the pathway is formed, next step will be substrate level phosphorylation. And substrate level phosphorylation is the synthesis of ATP from ADP using this high energy phosphorylated compound as a donor of phosphate group and as a driving force, as an energy resource. So once we transfer this phosphate group from 1,3-bisphosphoglycerate onto ADP to form ATP, what is left from that structure is 3-phosphoglycerate. So you can notice here that the carboxyl group of 3-phosphoglycerate is actually the same carboxyl group that we are looking for in pyruvate, right? And remember the rule, once a part of the molecule, a part of one of the intermediates is the same as this part in the product, no changes will happen at this part of the molecule. So it means that that carboxyl group will need to stay the same, and the remaining changes need to be performed here. And what should be the changes? So this carbon should become a keto group, right? Part of the keto group, and we need to remove oxygen. And the best way how to do it is to actually isomerize 3-phosphoglycerate into 2-phosphoglycerate. So you see, we kind of move this phosphate group from the third hydroxyl group to the second hydroxyl group. So now we can easily remove this oxygen atom, which is absent in the product, through dehydration. So we are going to remove this OH group together with this proton to form a double bond between those two carbons, right? So we remove water. This process is catalyzed by enolase, and we will form phosphoenol pyruvate. This is another high energy intermediate in glycolysis, actually the second high energy intermediate formed in glycolysis. So again, in order for us to know what will be the next step, we need to recall those rules, right? As soon as a high energy intermediate is produced in the course of the reaction, in the course of the pathway, next step will be substrate level phosphorylation step. So this is exactly, is what is happening, right? Another ATP molecule is synthesized, and finally pyruvate is produced. This step is catalyzed by pyruvate kinase. Okay, I have a question for you. What is the net energy gain when 1 mole of glyceraldehyde 3-phosphate, this guy here, is converted into 1 mole of pyruvate? So we're just looking at the payoff stage of glycolysis. Please pause the video, think as long as you need, and I will provide the correct answer in a second. So in this case, we are not investing any more ATP, right? So the first step will be our oxidation reduction reaction that would generate one NADH molecule, and then subsequent step will give us one ATP as a result of substrate level phosphorylation. And finally, the very last step of glycolysis will give us the second ATP. So the correct option would be D. So 1 mole of NADH and 2 moles of ATP will be produced. But what if we are talking about the net outcome of glycolysis, when we start with glucose and form pyruvate? So please pause the video, think as long as you need, and then I'll provide you the correct answer. So if we take into account both the investment stage and the payoff stage of glycolysis, we need to take into account that payoff stage of glycolysis needs to be taken twice, because we need to have 2 glyceraldehyde 3-phosphate molecules going through the payoff stage. So as a result, we invest 2 molecules of ATP at the investment stage, plus we synthesize 2 molecules of ATP per each glyceraldehyde 3-phosphate, as well as 1 molecule of NADH per each glyceraldehyde 3-phosphate. So as a result, we will produce 2 moles of ATP total and 2 moles of NADH. And this can be seen here, right? Again, investment stage required 2 molecules of ATP to be consumed. Here we generated 1 NADH, but we for 2 triose phosphate molecules it would be 2, right? And those 2 ATP molecules will be also doubled, right? So we'll produce 4 total. And then we take into account the investment, the cost of the investment stage. The net outcome will be 2 ATP and 2 NADH molecules.